So I
perfect.
started recording, and
Okay.
whenever you're ready.
Okay, that's awesome. Let's go here. Let's see if I can do present mode like that. Are you able to see that opening slide? Yeah, okay, great. Well, welcome everyone and those watching remotely. My name's Ray Cinti. I'm a biology teacher for several years, although I'm more of a student than I am a teacher. As I tell my students, this is a little intro to who I am. I think teaching ultimately, this is what I've learned over the years, is that it's really modeling learning. And I think if you're with the students, investigating and discovering with them. Of course, you have expertise, but if you're with them, they'll go along with you, and as opposed to telling them this is what you need to know. They're impressive this way. So I teach at a school down in Waitsfield, Vermont, called the Green Mountain Valley School, very appropriate to Vermont. It's a ski academy, and I teach biology down there. And I also teach anatomy and physiology at the Community College of Vermont. And I also am a fellow with a biotech company in the San Francisco Bay Area called Bio-Rad. And their particular reagents are the ones that I want to talk about today in terms of how you can involve students in not only the knowledge, but actually a hands-on investigation using real antigen and real antibodies to do a COVID rapid test. I can't think of anything more relevant and current than this. I mean, this is on every student's mind. And so basically the title of the presentation is Detecting SARS-CoV-2 using an assay called enzyme-linked Immunosorbent assay, and so thus the name ELISA. And we call it rapid tests now. And so let's see if okay. So there's an attendee. The company Bio-Rad likes to gather information about participants in order to send information if you're interested in any of the reagents. And so I'm going to just shoot down a link there in the chat if anyone's interested in that. So coronavirus, what's in a name? Well, coronavirus, or COVID, is a large class of viruses. And so I thought I'd talk a little bit about the background just to pull everybody in, and then get in more particulars about the particular tests or assays for ELISA. So it's a virus, a non-living particle that can cause infection in humans and other animals. And the name, you might know, corona, comes from the fact that, as you can see in this transmission electron micrograph, that there's spike proteins that sort of look like a crown to individuals who first noticed that. And so the SARS part of this is severe acute respiratory syndrome. CoV-2, again, is the strain causing the current pandemic. And so COVID-19 is really the set of symptoms that's caused by SARS-CoV-2. And so, the importance of knowing what you're talking about there is sometimes crucial to students. I found a site on that I wanted to welcome everyone to. You might all be familiar with the Howard Hughes Medical Institute, or HHMI. They have a variety of resources on a lot of topics, and surprise, surprise, they do an excellent job with COVID as well. And so they have an animation. If I were to click this, I'm worried that something might go weird, but I'm going to click it anyway on this. And so when you click this, they have these short animations, and I just want to show you a little clip of that just to give you a sense of it. They have broken up COVID into different modules. This one's on infection, and then detection, and vaccines.
This is really the virus SARS-CoV-2. Its name stands for Severe Acute Respiratory Syndrome Coronavirus 2. You may have heard it called just the coronavirus or COVID-19.
So it's kind of the things that I was just saying. They're great links and they're available on the internet. And I find myself either showing them in person to my students or sending it out ahead of time so that they're able to watch something prior to class, so that class is more of a discussion or conversation as opposed to me telling them information, because they're certainly capable of watching something like that. So as far as detection of a virus, there's a variety of ways in which the virus can be detected. It's kind of fun. I don't know if any of you agree with this, but usually something like PCR, polymerase chain reaction, was something that was only a discussion pretty much in a life science class, but now everyone on the street's talking about PCR, which I think is kind of fun. Samples are taken usually from the nasal cavity of a patient, and then the virus is then treated with, it's a retrovirus, and so it has RNA, and so it's treated with an enzyme called reverse transcriptase to turn it into DNA, and then using primers you can look at a particular area that would identify the virus, and then PCR is basically just DNA amplification, and you can detect whether or not the virus is there. This is the gold standard in identification, but it's kind of costly, truth be told. And there's an interesting story about why we started working with PCR and not the ELISA. But the ELISA is a more readily available test. I'm going to say it's fairly reliable if used properly over a series of days. And you can test either for antigens, which is the protein itself that surrounds the virus, or the spike proteins, or you could look at something called polyacrylamide gel electrophoresis. In other words, you can look at particular proteins of interest and separate them in a gel. And there's also something called Western blot. And for antibody detection, again, ELISA is an indication of that. So this presentation is going to be a talk about antibody detection and antigen detection. And so, as I was mentioning before, reverse transcription PCR or real-time PCR is viruses are then lysed. Their viral RNA is released. Reverse transcriptase will turn it into DNA or cDNA, and then primers are added, and there's a particular segment of that DNA that is then amplified. And depending on what kind of thermal cycler is being used, you could do real-time PCR, detect it using fluorescence, or you could actually do endpoint PCR and actually run a gel and look for those particular amplicons. Feel free to ask questions or just interrupt at any time if something needs further clarification. I don't normally like to talk quickly or over the heads of students, but I'm assuming a level of understanding of this. So these ELISA tests are proteins, virus proteins, otherwise known as antigens, and we can detect those. And so therefore we can detect the presence of the virus in a patient. So we can detect those spike proteins, as you can see in these pictures right here. And we can also detect the antibodies that a patient produces against the virus. And so those are both ways to determine whether or not a person currently has the virus or had the virus. So pre and post past tense. So what you're seeing here is a light microscopic micrograph of red blood cells. This is plasma in between. And these fellows right here are a type of white blood cell called a lymphocyte. Now, all white blood cells are really important, but I want to do a little shout out to these guys, these lymphocytes, because lymphocytes can produce a class of gamma globulins that circulate in the plasma of the blood and the lymph called antibodies. And these antibodies are really useful because lymphocytes will produce them and they'll circulate, and they can bind to viruses and also bacteria and neutralize them in a variety of ways. Antibody biochemistry is kind of interesting, but they produce antibodies that will fight against viruses. Okay, And so a close-up of a serum antibody. Serum is another way of saying plasma when it's pulled out of the body. I like to tell the students that an antibody kind of looks like a lobster. It has like this body and these arms and these claws, and there's hundreds of thousands of different antibodies, and each of them are specific to a particular antigen. And so this is all the strength of our immune system and also a little bit of a weakness, if I might say, because we have the ability to produce antibodies against all these viruses. We can survive. However, they're in low concentration. And so when something comes along, gets us sick, like SARS-CoV-2, and it's particularly worrisome if it's a very dangerous or acute virus, it can cause us to be fatally sick. And so our body generates these antibodies, but it takes a few days and even up to a week or 2 weeks, as you may know. But what happens is that it attaches to an antigen and neutralizes it. And so antibodies will circulate in the bloodstream, and we can detect those. And they're very specific to every particular virus in general. And so what would be normally done in a medical clinic is a phlebotomist, a nurse, or a doctor will take a sample of a patient's blood and spin it down, centrifuge it, and here's the plasma, which is about approximately 50% of what the blood is. And it's in here that you would find the particular, if I do this is where you would find the antibodies in the plasma. So that could be separated and then used as part of the detection mechanism. And so you wouldn't find just the COVID antibody. All of your antibodies are in there. And so this is where the cleverness or the intrigue comes into when it comes to ELISA. Like, oh my God, how can you do a test? How can you do a rapid test that would look at an antibody that was just specific for COVID? I mean, it's quite remarkable, but that's the whole story with antigen and antibody tests. And again, this particular link, I could share this PowerPoint with those of you who are watching as well. But this particular HHMI website on the biology of SARS-CoV-2 has a really cool animation on detecting COVID, but we're about to talk about it as well. Briefly, although this is of interest, when you look at the infection timeline, a lot of discussion on NPR and news channels about this. Doctors are weighing in on this as it relates to whether or not the rapid test is capable of detecting and false positives and false negatives and how it is. And so just wanted to bring this to your attention. So when a virus enters your body, the antigen itself, if you're looking at the red, in the first day or two, and third day, the antigen of the virus material is very high. And so PCR would be a great way to detect the virus at really any time, because PCR is extremely accurate and it would be able to look at the viral sequence and detect it. Now, an antigen test is also reliable, especially if you take it on the second day, the third day, the fourth day. In other words, taking it over a few days, because the antigen is at high concentration. It's almost like a bell-shaped curve. What can happen, though, is when the body defeats the virus, the antigen itself goes down, and the ability for the ELISA test to detect it is not as good as it is in the first few days. Now, when the body produces antibodies, the antibodies can be detected also using ELISA. And so there's two different kinds of tests. There's an antigen test and an antibody test. You can think of it this way. An antigen test is trying to tell the physician if you have the virus, and the antibody test is trying to say whether or not you had the virus or not. And so I'm using the term antigen, but the antigen is, in this case, for SARS-CoV-2, the spike protein. this is an area that's specific and it's a protein. The word antigen means antibody generating material. And so it's a foreign substance that triggers the immune system. So the CDC recommends using the spike protein, and that is what is used as the antigen in the ELISA test. Now, the ELISA test itself can be performed with students. And so, and it's not simulated, it's real antigen, real antibody, but not of COVID. I'm going to say, of course, because that would be very dangerous, but antibodies nevertheless an antigen. And so there's a really interesting animation that I want to bring to your attention. And let me see if I can click on that and show you. And so you would give students microplates, which are these little tiny plastic wells in front of them. And what you would do is, this is going to walk you through. This is what's known as the antibody test. And so this is also known as an indirect ELISA, because you're trying to determine whether or not you have the virus based on whether or not the patient has produced antibodies against it. So the very first thing that you would have students do is pipette antigen into the microtiter plate, and then that would adhere to the walls of the vessel like this. And then what you would do is you would wash using just a phosphate saline buffered solution, basically a detergent that washes out any unbound antigen, and it also covers the surface. And then what you would do is you're trying to figure out if the patient has COVID or not. So then you would introduce the patient's serum. And so the serum, as you may remember, contains antibodies, lots of antibodies. I mean, we have antibodies against all the diseases that we've had in our lives. And so we're like, oh my God, how's this going to detect whether or not I had COVID or not? Well, if you had COVID, it would actually, your antibody would actually specifically attach to the antigen that was present when we first put that into the microtiter plate. As you can see in gray, look, here it is attached. But you wouldn't be able to see that if you're performing the test, because these proteins are, of course, clear and they wouldn't be visible. And so what you then do is you do a second wash, and I'll go through some of the steps that you would use with the students on this. And so you'd wash that out. And so any unincorporated antibodies would be released. Now, this is the kind of, I think, the cool part of this investigation. In the lab, biotech companies and pharmaceutical companies have been able to create a secondary antibody, shown here in green, that actually is specific to the primary antibody. So it's an antibody that's going after an antibody, and it's known as the secondary antibody. And so when you add that, it also has an enzyme linked to it. Do you see this thing that looks like an orange Pac-Man right there? So this is the namesake of the assay, which is enzyme-linked Immunosorbent assay. And so this is the key event. And so you're like, well, what's this doing? I still can't see anything because these are all, again, clear. And then, okay, so this is also called a colorimetric assay, meaning that the reason that enzyme is there is that when you add a substrate, the enzyme will speed up a chemical reaction and break down the substrate, causing a visible color change to occur, as you can see in this final step right here. When the enzyme interacts with that, it'll turn the whole solution, whatever color it'd be, yellow or blue, and bam, you would see it. And so this is how you would detect. And now you could do this with a person, so you can see this with your eyes, or you could send it to a machine that will shoot a laser at it, something analogous to a spectrophotometer that would be able to read this, because there's so many samples being done. So that was kind of quick. And so I'm going to now transition into what you would do with the students. You would say, okay, the first thing we're going to do is we're going to add a viral antigen. So this is the surface protein to the microtiter plate, and that the antigen is going to bind. And they would say, well, how's that going to happen? And it's like, well, the protein spike, this is just a small showing 2 amino acids are adhering to the plastic. This is the plastic of the microplate. You would create like a scenario to sort of make it realistic. You'd say, okay, listen, we're working at the lab. You know how we're taking these rapid tests? Well, there's a lab that also creates these rapid tests, and they also do PCR. And so we're gonna be those medical lab assistants and take care of this. And we're going to try to determine whether or not a person has had COVID or not. And so you would list with the students what some of the reagents are in front of them. A funny thing, I was listening to NPR the other day, and that there was a doctor talking about various reagents, and the host interrupted and said, you know, can I ask a question, like, what's a reagent? And so that's a reminder that, you know, not everyone knows the terminology, but the reagents are all these chemicals or proteins, mostly here, that we're talking about. And so what you want to then introduce to the students, that in order to validate whether or not the reaction is working, you have to use a control. In other words, you have to have actual antibodies that would show the patient was positive. And you also have to have a negative control, which shows definitively that the patient is not positive. And if positive is positive and negative is negative, then the kit is more reliable. Like when, for example, like when you purchase a home kit like this, you'll know that there's a little pink line that says whether or not the control is working or not. And then you'd remind the students, okay, the antigen is the spike protein. The secondary antibody is the one with the enzyme that connects to the primary antibody that you're looking for. The substrate is what the enzyme is reacting. And then, of course, the patient's serum. And so, making it a little bit fun, I always like to put the names of the students on the micro test tube to see whether or not, you know, a particular student is COVID positive or negative. I also, if you're familiar with it, you'll know that the ELISA test can be used for other things too. Like, for example, the detection whether or not a person's pregnant or not. And if you've ever taken a pregnancy test, the same sort of rules apply. You have to have the one line suggestive that the test is working, and then if there's a second line, then yay, you're pregnant, or yay, you're not pregnant, depending on the situation. So the first thing is you're going to add the antigen. And so you're like, well, how do I add a liquid to these little tiny test tubes? You can use a little transfer pipette, a plastics disposable transfer pipette, or if your particular school has my adjustable volumetric micropipettes, the volume would be 50 microliters, or basically 1 drop. And so you'd put the spike protein into all of the samples. Do you notice here in the picture, these are 3 positives, so it's triple to make sure that there's no spillage. There's 3 negatives, and then A would be 1 patient and B would be the other one. Or so 1 student or 2 students can do that on the same microtiter plate. This is what it looks like when the antigen is attached to the microtiter plate. And then you would simply add a wash buffer to each of those, and you dump it out, and then you on paper towel, and then you'd proceed. So washing is important, as I may have said this before, because it removes any unbound antigen, and it also blocks the rest of the surface surrounding it. So at this point, once the antigen has been added, then you're going to add all of the other samples, and it's 50 microliters for everything, or 1 drop. And so you're going to add, do you notice here in the pink tube, this is positive because this is the antigen, I'm sorry, this is the antibody that we're looking for. So the key is, if a patient has this antibody, that means that they were exposed to COVID. And so therefore, this would be positive. The negative just has a buffer in it. And the patients were kind of curious, you know, do they have the antibody or do they not have the antibody? And so 1 student would go into A, the other student would go into B, and then you wait about 5 minutes. So this could be performed in a typical 40-minute to 50-minute class period. What's interesting about it, though, is it's fun to sort of drag things out. And what I mean by that is like really create the scenario the day before, then perform the investigation. And then the follow-up day is really like, you know, these are the day before, the day after are sometimes most important because it's designing the experiment. And then the third day, it would be like, okay, where can we go from here? What's going on? You know, like applying it to real life scenario, let the students be able to talk it out. And so when the patient's serum is added, if they had COVID, their antibody would attach to the antigen, even though you wouldn't be able to see it, but it would be there and it would be attached. Then you do a second wash, and then you add the secondary antibody. If you recall, the secondary antibody is the one with the enzyme conjugated to it. And so it's going to adhere to the primary antibody, again, not visible. The secondary antibody, as I was saying before, is able to attach to the primary antibody, and it has the enzyme. And it's what is going to help us detect what's happening. And so this final wash is maybe the most important wash because you want to be able to wash out the secondary antibody if a patient is negative, and if a patient's positive. If a person's positive, the secondary antibody will really be sticking hard to the primary antibody and to the antigen. But you want to wash out any unincorporated secondary antibody so that it doesn't give a false positive. And then the final step is to add a drop of substrate. And then in just 2 to 3 minutes, 5 minutes max, it'll give a color. And usually the color is the result of the enzyme fitting into the active site and then changing color into blue. And this is what it looks like. And so you can see here, these are all positives, these are all negatives. And then, oh, this student is positive, this one's negative, this one's positive, this one's positive. And so that's always kind of fun. The background behind this, I said it before, but just to sort of walk it through one more time. So, you know, the virus is what's infecting a person. And so the virus is this antigen, and the person makes antibodies against it. And what we're trying to find is that antibody. We're trying to look for that. And it's like, how do we look for that antibody? Well, we add a purified antigen to the well. And this could be purchased at a biotech company or pharmaceutical company. So you put in the antigen into the well first. Then you put the patient's serum in. And if the patient antibody is present, the second antibody will attach to the primary antibody. Your question might be, where does the lab get secondary antibodies? And the answer to that is, interestingly, that there are other animals that can come down with SARS-CoV-2. And if you, in fact, for example, a goat with SARS-CoV-2, they will produce antibody against it. So that's the primary antibody. In the lab, we're able to make, like artificially make, although it's real, a secondary antibody that is specific to that goat antibody. And so that's how we're able to do that. And so this is how it works for people too. That secondary antibody will be able to detect human primary antibody. Any questions on that? No? Okay. So again, positive would be blue and clear would be not. So it depends, you know, if you're detecting a virus that's unhappy, if this is a pregnancy test, then maybe these smiley emojis would be reversed. And again, I showed this before, but I want to transition a little bit between what is an antibody test and an antigen test. An antigen rapid test is going to detect the presence of the virus. Do I have COVID in my body right now? Have I been exposed to the antigen? We can actually go after the antigen as well. And it's extremely reliable, especially if it's used on the second, third, fourth, and fifth day consecutively. After that, it becomes less reliable because the viral load is decreased when the body begins to fight it off, although PCR would still be effective. So an antigen test is a way that we can try to find whether or not the antigen is present. And so the way that works is that we would expose a rabbit, and this is traditionally, it may be a different animal, but this is traditionally how it's used. You expose a rabbit to the antigen, and it would produce a primary antibody against the antigen. And then the primary antibody is then used in a goat, and the goat would produce the secondary antibody. And so what would happen is we would put a patient's serum or a Q-tip swab into a solution. And what would happen is we would then we would introduce primary antibody and secondary antibody, and that would detect whether or not the antigen is present or not. So it's similar, but slightly different. This is also an animation using an antigen detection. So you're trying to determine whether or not a person is positive with the virus currently. This is also known as a direct ELISA because you're going after the virus direct. And so you might be familiar with this because the home kits, sort of like a home pregnancy test, instead of having, can you imagine having like everyone at their house trying to pipette and in a microtiter plate and then wash buffer? That would be a really complicated. And so at the lab, we use lateral flow devices or as a lateral flow immunoassay. And so what's totally remarkable is that when you put the patient's sample in the little hole, and then you add a buffer using capillary action, the reagents migrate up the nitrocellulose strip, and they begin to do each of those steps. In other words, they would reach the primary antibody, the secondary antibody, and the substrate. And if it's working, it'll give one line, and if you have the virus, it'll give you two lines. And so there's, again, a lot of different companies that are producing these rapid tests. And they're becoming, the government is getting very involved in this, as if you're keeping up with current events, and they're making these tests available for free. Although whether or not you can get them is always, the challenge because of the distribution and the pandemic nature of this is kind of overwhelming. But I sense that everyone on board is trying. There are websites where you can direct the students to go if they're wondering, like, this is a particular link right here. Let me just click it just for a second, just to show you. It's made by the company and it's on YouTube. And when you click on this, it goes step by step, showing you how to perform the test and how it works. It's pretty cool. And so you could do that with the students in conjunction with actually performing the experiment. And then back to sort of backing away, like concluding with the workshop, what's a real important concept is the fact that the virus mutates. And so this is an opportunity to bring up with the student how when nucleic acids, RNA or DNA are being replicated or synthesized, errors occur, especially in RNA viruses more readily, so they don't have a proofreading enzyme. And so when mistakes occur, that results in a new novel protein or a new variant. Whether or not that's good, meaning that in the human perspective, if there's a protein that becomes inefficient at binding to human cells, that's good for us. But for the virus perspective, maybe not as good. And so there's been a variety of variants that have been making the news, of course, Delta and Omicron. And so there's a really cool link on Howard Hughes Medical Institute on the making of new variants. And boy, I can't think of anything more important than this discussion leading the students into what is at the heart of natural selection, which is random mutation, which creates variations in a population, and then nature selects which ones are more fit, meaning which ones are capable of reproducing better in the long haul, and then those variants become dominant. I mean, that's pretty much the heart of biology. And then again, another opportunity to get into a conversation about vaccination. How does that work? What's in the vaccine? Messenger RNA. In my mind, this is one of the great positives of this pandemic, that we have these RNA vaccines, and how they're created and how they, the details. If you're teaching an advanced class, when you're talking about a 5' methyl guanine cap on the front of it and a poly A tail, so that the messenger RNA stays longer in the deltoid muscle, so that it's able to express the spike proteins, and how helper T cells can bind and then communicate with the lymphocytes to produce the gamma globulins. It's such an opportunity. There's even websites that'll show the sequence of the messenger RNA in the vaccine, which also shows that there's particular sequences called signal regions that will allow the protein, the spike protein, to be produced on an attached ribosome to rough endoplasmic reticulum and smooth ER, so that those proteins can manifest to the outside of the cell membrane of a muscle cell. It's all very interesting. Again, current events, this is an opportunity to get students in the habit and the routine of listening to podcasts on the radio, reading the newspaper, introducing various articles from journals that are coming out. But there's a really cool NPR just the other day about an MIT researcher who was working with ELISA for other particular diseases, and quickly created a rapid test in the spring of 2020 that would have been able to detect COVID, and it would have been a game changer. And so this is on point NPR. You can look it up on January 13th. So just the other day, basically. Fascinating conversation about that and some of the politics behind why that wasn't happening. And then, in conclusion, where can I find these reagents? You can order ELISA or rapid tests really from any pharmaceutical company, but I'm working with the company Bio-Rad, and they have a division at the biotech company for life science education. And so if you went to biorad.com, you'd be able to find kits that contain the microtiter plates, all the micro test tubes, pipettes, the wash buffer, the antigen, the primary, secondary, and substrates to perform an ELISA test with the students. It's an opportunity to let the students have a hands-on experience with rapid tests. And when students participate, if I could just say this, when they participate in this level, what's going to happen is they're going to be proud of their learning, because it is complicated and it is relevant and it is important. And what they're going to do is they're going to go home and they might share this with their parents, or they might share it with their friends. And this is how it begins. This is why, when I, as a childlike as I am, and I said at the start of this, I'm more of a student than I am a teacher. Whenever I'm discovering something or working on something, I'm first person home telling my kids or my wife, guess what? So this is the joy. It's relevant. They're participating. It's something that's overwhelming for young kids. You could slow this down. I don't teach elementary school, but if you wanted to do this type of rapid test for like middle school, I think it's all possible if you just slow it down and and meet the students where they are. And if you show passion and you show like this is really important, and everyone needs to really buckle down, and this helps the students overcome sort of, I worry personally about the anxiety that students feel these days. And I think knowledge in some way helps. It's not everything isn't out of their hands or out of their control, and it's not all these the adults who are making all these decisions. I mean, the kids could be really excited about being able to perform a rapid test in their classroom. I think it's pretty cool. How about that?
Thank you. I know Adriana asked a question in the chat. I'm going to call her out. And I'm going to have her ask the question.
Yes.
So yeah.
I really enjoyed this, Ray, a lot. I am a scientist by training and an educator at heart. So I really appreciated how you went through all the steps of what goes into an ELISA, because I was thinking back like how I taught ELISA to folks, and I saw a lot of mistakes in my teaching, watching you go through it. So I was like, oh, they could have benefited from understanding like how the secondary antibody is like modified, and you can attach different substrates to it, how the colors come to be, because it was always a little bit mystical.
No, truly. And again, I'm a fan of slowing it down with the students, because like if you let them, tactile, if you create models and paper cutouts and show white blood cells and show like just really slow it down, and it's an opportunity to stay with one thing and walk all around it. In other words, protein synthesis or replication, PCR, vaccines. I mean, basically this one investigation, there's a lot of ways in which it could start a conversation, or it could be a way to revisit something before. Because sometimes, when you're going through basic biology, if you're talking about protein synthesis, and it's like, or natural selection. Natural selection isn't just back in Charles Darwin's time. Like this is natural selection. It's right in our face.
Yeah, that was related to the question I put in the chat, because I'm a developmental biologist, and I love talking to kids about, like, I ask them like, so how babies are made, and they always get the giggles and they're like, ha, and they try to explain like the mechanics of it, and I'm like, no, but what happens after that, and guiding them through that process. So I rely a bit on shock factor for that specifically, but I wanted to see your approach of how you talked about natural selection with kids. And I work mainly with elementary school kids, so that's why the shock factor works. But sometimes I've applied it with high schoolers, and they're like, "You're cheeky."
Yeah, and I hyperbole alert, but what's more important than that? I mean, like, there's so much misconception about that, and so much controversy with this concept of evolution. But this is evolution. This is a change over time that is occurring because of a mistake, and there's a different protein, there's a different variant. It's right in front of them. I think students will accept it. And the national standards really, I'm a fan of all of that, because it's all about when you're making a claim, the evidence to support that. I mean, that's the heart and soul of science, right? I mean, this is it. This is the evidence. That's these variants. I mean, if this is what it takes to sort of get that concept into some minds, so be it.
Adriana, I hope that you're going to be with us tomorrow when we do evolution for elementary school. That's coming up tomorrow. Ray, I was wondering, just with the nature of like the sensitivity of like testing for COVID in schools right now, and like what would happen if a student had a positive test, are there opportunities either for something to be synthesized where it's more of like the students aren't testing themselves for COVID? Like, like, and I think specifically to like the animals, like could an educator be able to take like animal samples and create a simulated environment in the school, just because of the, like, I hesitate where I'm, and I totally get it. Like kiddos are taking the antigen test, they're taking the PCR test, but like, I just wonder what the fallout would be if if a student actually tested themselves for COVID and it turned out to be positive. Do
Yeah,
you
no, I
know, like,
agree.
does
I agree.
the lab like send synthetic stuff
It,
out, so it's a
it,
little bit more
it
like,
does. It
okay.
does. Those particular reagents, though real, isn't
Right.
not using the SARS-CoV-2 spike protein antigen. They're using a different antigen that's sort of proprietary to the company Bio-Rad. But nevertheless, those are real antibodies against they came from the rabbit, and the secondary really came from the goat. And they're able to reveal the enzyme is I think it's horseradish peroxidase. But those are all real reagents. But if you were to order, this is the kind of funny a little bit to your point. The same company Bio-Rad will create these like educational reagents, but they also sell the real ones. And I would not, just opinion, I would not do that with the students.
Yeah,
because
no, there's a lot of trauma that can come from that. And
yeah, of course you don't want to like alarm anyone or stir anyone, and it's emotion, and it's very important because they're for a variety of reasons.
Right.
But I think it's relevant, and I think it's important, and I think when the students are made aware of what is actually going down, it takes away a lot of the magic and mystery, gives them a little bit more ownership of something that is so
well, that's what I
scary.
really appreciated. But even just as an adult, having you walk through it, where I was like, oh, I knew all this, but I hadn't seen it laid out in a way where I was like, oh yes, like I got it.
Yeah.
My other thought is that those simulations that you were showing earlier, the one I'm trying to remember, like it had those are those, how do I want to ask this, like, can an educator use those for like an online or hybrid or virtual? Because as we have schools that are like trying to figure things out, like I think of that and I'm like, that's a great opportunity to bring in like virtual learning if if a student is either on like a flexible pathway or if they have to be at home quarantining or whatever.
So to speak to that, look, so all of it can be converted into a virtual thing. Like you could, even things like PCR and electrophoresis and all of that, you could just walk students through and what's going on, and then show them pictures of results, or show them a gel, and they have to figure things out from banding patterns, etc. And so that being said, of course I'm biased to it's more impactful when you actually do it. It's more impactful when you're actually pipetting and you're spilling stuff, and you're confused. More like it, you're confused in what the step is, and you have to consult with your next door neighbor in class, and your neighbor becomes an ally and someone who you can collaborate with. And then that person's self-esteem goes up because they're feeling like, wow, I just helped my partner over here, and I feel good about that. And then that's the real lesson I've found over the years teaching, is like collaboration, making mistakes. So the virtual, so that's a long response, but of course all this can be done, but it's less mistake prone.
Absolutely. There's a lot
the
of
kicker.
learning that
That's the
happens.
kicker. Like, yeah, you could like just a quick thing. Like, you
mistakes.
could think that you're a good cook by watching like tasty videos on YouTube, like I do. But then when you're actually in the kitchen doing it, you're like, oh my God, like stuff going crazy. But that's
I
that's how
wonder if there's a way to merge that virtual though, because if, rapid antigen tests and like PCR, like, well, not necessarily PCR, but like if you can, like the test at home, those kits,
yes.
being able to say, okay, your students are virtual and remote. However, we're all going to be using the same kit or whatever, then everybody kind of does have that experience. I don't know. I'm just
Well,
like just thinking out loud.
And some of the scientists at the company Bio-Rad, we have these conversations about like what can be sent home. Like, is this where we're going? Where like, a box arrives at the house and the kids open it up, and then you go online with the students and you go, all right, listen, everybody open up their stuff. Okay, everyone grab this chemical and add this. And so everyone's doing it, but at their own location. And it's hands-on, but it's happening at a different location. And this is where some of the chemicals are maybe not as safe as others. And the lawyers get involved. I'll just say that we're things can get, but the simulation can work. Like, me personally, like I try to create a lot of educational videos that I'll upload on YouTube, and it's like, oh, you're explaining things that only can go so far, because unless the student wants to know, it's like what we were saying in the previous with the keynote speaker. It's like the key to education is to not give them the answer to the question that they never had. It's like you have to create an actual authentic inquiry, a mystery. It's the story arc. No one wants to hear about the end of the movie right at the beginning. You have to create a struggle, and there has to be the hero. They're the hero, and they're climbing, and the teacher is like the person who's going with them to fight the beast. And in this case, it's SARS.
So,
and
I'm sorry. I was just to say, Adrian, just for the sake of time, Adriana has, I think, 2 questions in the chat.
Sure.
Sorry.
Since you're more familiar with the Bio-Rad life sciences materials, can you just speak which of those are really popular? So I can forward them to my group. I'll let them know that they can purchase these. And then the second thing is that you talked about like virtual labs and how kids are like in the virtual, if they're over here, how are they going to do labs at home? Has Bio-Rad looked into developing like kitchen science labs for students to home?
Tough to hear that. See, by trade, kitchen science labs. Has Bio-Rad developed more materials that lend for virtual labs? They have. There are some available at the website. If you go to biorad.com, there's things like that you really can't harm yourself, like things that are really important. Extraction of DNA modules. They have things that are kind of interesting there, and a lot of the focus has been at Bio-Rad is to create authentic, real life. That's the mission statement, is to create real, because it's really a biotech company. And there were a few individuals in there that believed that, when students actually participate in real science, that's going to be the key, and making sure that it's relevant. And so, yeah, there are some kits that are available. The truth is anyone is capable of ordering any of these things online. Nevertheless, that's.
And did you, and her first question, and Adriana, you can correct me if I'm wrong, was what Bio-Rad life science resources are really popular.
Oh yeah, and they have a lot, like those ones that I kind of walked you through in the presentation, where step by step what's going on. What they've also created most recently is, and they've placed on YouTube, is video of in that's time lapse. So showing like the steps of the lab visually so that students can follow it, and the methods, like the steps. They've also created, if you've ever seen these videos where there's like a hand drawing animated cartoons on a whiteboard, and it's sort of sped up, they've also created those kinds of videos to help students. And so I love that, because I'll have the students like on the screen, I'll have that step being shown on YouTube, and then we'll talk about it and I'll put it on the whiteboard. It requires a kind of a multimedia thing. So yeah, tremendous amount of resources. But I use, like, in addition to Bio-Rad, I use resources from, a variety of places, as well as making my own. And then, you can even get students involved as a project and say, listen, can you create a podcast interview? And, you're the scientist and you're trying to explain a rapid test to the principal at your school, or get them involved in those kinds of issues and get them to take different perspectives. Like I find it kind of fun to go after it, like, and say, well, what is the controversy about this? Like, let's get a debate about it, and force people to take a different alternative perspective and see what and put themselves in the shoes of what someone else might feel. And then that creates authentic empathy and understanding, and like, and what's going on, instead of just coming up with a wall, like, oh, these people don't understand the goodness of a vaccine. Of course, but what is it that's troubling, and why? And if you can create those kinds of debates in class in a controlled environment, then they're better able to then, when they leave, be able to actually have those conversations when it matters, potentially. But doing the lab, it's a lot of fun. This again can be done for a pregnancy test too, and that creates a completely different scenario. Like, like, oh my god, But that's what it is, a rapid test. And these rapid tests can also be used to detect proteins, genetically modified crops. You could use it for a variety of things.
Right. Well, thank you. It was fascinating. Like, just really cool to go through the process. Like, it was neat. I like it.
Well, thanks everyone. The last thing I wanted to say is that, as we as a teacher, we go to, we have degrees that we've obtained in college, but I think constantly going to conferences like this, sharing ideas, or just learning on your own and sharing it with the students. I think the students always like raise an eyebrow when I tell them, guess what I learned the other day by reading this, or guess what I found out the other day when I listened to this. And what's really interesting? I don't know this. Like, I start off class like, this is what I don't understand. And they're like, what do you mean? I go, well, I don't understand it. Now I'm really interested. It's like, I'm not interested in something I already know. That's the nature of science. That's it. It's like, a great scientist isn't great because they know things. It's that they want to know things. It's a big switch.
I know. So what do you mean you're not all knowing?
Yeah.
All right. Well, I'm going to stop the recording.